In this lecture, we'll learn about methods that are currently used to study microbial communities. There are two main challenges associated with studying microbes. First, it is difficult to distinguish microbes using morphology, since they come in a limited number of shapes. Take a look at these two bacteria. Do you think they're the same? They might look similar, but the one on the left is Salmonella, a familiar foodborne pathogen, and the one on the right is Mycobacterium, which can cause tuberculosis. The second challenge with studying microbes is that the majority of microbes, especially gut microbes, cannot be easily cultured. Many of the methods Norm discussed in the last lecture require us to grow the microbes to study them. Using DNA to identify microbes solves both of these problems. As a result, over the past 2 decades, microbial research has become increasingly dependent on using DNA to examine microbial communities. DNA, or deoxyribonucleic acid, is a molecule found in humans and all living organisms that stores information about our bodies and how they work in a code. This code is a sequence of 4 chemical bases or nucleotides. They are adenine, or A, thymine, or T, cytosine, or C, and guanine, G. DNA is arranged in 2 long strands that form a spiral called a double helix. In this ladder-like arrangement, the nucleotides are like rungs that connect one strand of DNA to another in a special way. Adenine bases can only pair with thymine, while cytosine can only pair with guanine. This means they are complementary to each other. The word genome refers to all the DNA in any one living organism. The genome is like the blueprint that your cells use to make the machinery they need to run. This picture shows the human genome printed out. Each letter represents a nucleotide in our DNA. Within each organism's genome, the DNA is organized into genes. Genes are regions of the genome that correspond to a unit of inheritance, such as blood type or hair color, although these traits are actually determined by multiple genes. A gene is like a single part of the entire blueprint of a cell. To pass on their information, genes are transcribed into messages and translated into proteins by our cells. Proteins do most of the work in our bodies. They are the machines that the blueprints provide instructions for. Some genes are the same in all organisms. These tend to be genes that control important functions that are necessary for a cell to work. Other genes vary between organisms and result in differences between these organisms. In microbes, these genes might control what compounds can be used as food to produce energy. Within genes, there are also areas that are the same or conserved among organisms, and others that are different or variable among organisms. The areas of a gene that are variable are the ones that drive the evolution of a species, or its change over time in response to the environment. Using DNA in microbial research depends on these conserved and variable regions of genes across species. Specifically, we look for genes that all microbes have and use variation in those genes to tell different species apart. This allows us to study microbial communities using their DNA so that we don't have to grow them. The 16S rRNA gene is found in all bacteria and archaea, and only in bacteria and archaea. Humans don't have this gene. The 16S rRNA gene is approximately 1,500 nucleotides long. However, like most genes, it contains regions of conserved and variable sequence, some of which are more variable than others. We use the more conserved regions to pinpoint the gene, and we use the variable regions to tell species apart. Up until now, it appears that the V4 region is one of the best regions for telling different types of bacteria and archaea apart. So most microbial analyses target this part of the 16S rRNA gene. There are 4 basic steps involved in using the 16S rRNA gene to tell types of bacteria and archaea apart. First, you need to get the DNA out of the cells. We refer to this as extracting DNA. Then, you have to make copies of the part of the 16S gene that you want to examine, such as the V4 region. This is done using polymerase chain reaction. Then you have to sequence the copies of the DNA. Finally, you use the sequences to distinguish microbes from each other. Let's start by talking about DNA extraction. DNA extraction involves a series of physical and chemical methods that break cell membranes and let the DNA out. The material, in the case of gut microbes, fecal material, gets ground up and shaken with tiny beads, and we add things like salt solutions to poke holes in microbial cell membranes. We then use a series of additional chemicals and filtering methods to remove other proteins and contaminants that we won't use. At the end, we have isolated, clean DNA that we can use for analyses. Although it's not necessary, many people do DNA extractions using special kits they buy from laboratory supply companies. These kits include all of the chemicals, tubes, and filters necessary to complete an entire DNA extraction and speed up the process substantially. Now that we have the DNA, we have to make copies of the region that we're interested in. DNA extraction is not limited to one type of DNA. When you extract DNA from a fecal sample, you're getting DNA from the microbes, but you may also be getting DNA from the plants a person is eating or from other cells that are in the fecal material. We don't care about that DNA, so we have to separate it out. We do that by making lots of copies of only the microbial DNA and throwing away all of the other DNA. We make copies of the microbial DNA using polymerase chain reaction. Polymerase chain reaction, or PCR, mimics how DNA is copied in nature. So let's talk about what happens in nature first. To copy itself, DNA first has to unwind. An enzyme called DNA helicase does this. It basically looks like a zipper unzipping the DNA. Once this happens, another enzyme called DNA polymerase moves along each strand of DNA and adds complementary base pairs. Remember, only adenine and thymine go with each other, while cytosine and guanine also only go with each other. After DNA polymerase is done pairing these bases up, you end with 2 copies of DNA instead of 1. Each copy is made up of half old DNA and half new DNA. That sounds pretty easy, right? It is, but DNA polymerase has certain requirements it needs to work. It needs single-stranded DNA to serve as a template, a starting place where the DNA goes from being double-stranded to unwound and single-stranded, and free nucleotides, your A's, G's, T's, and C's. So let's talk about PCR. PCR is simply the laboratory synthesis of DNA from a DNA template. Just like in nature, PCR has similar requirements. We need DNA polymerase, a DNA template, and nucleotides. Remember, DNA polymerase also needs a starting point. For that, we use oligonucleotide primers. These are small pieces of DNA that we make or order from a company and add to the reaction. They show DNA polymerase where we want it to start and stop copying. This is what we use to target different genes or gene regions. For example, if we want to make copies of the V4 region of the 16S rRNA gene, we add primers that match conserved regions of the DNA that are located right before and after the V4 region. That way, DNA polymerase will find that region in all of the microbes and make copies of the variable sequence in between the primers. Here you see 2 pieces of DNA from 2 microbes. The conserved region is highlighted in red. This is where our primers would tell DNA polymerase to start and stop. The black letters represent the variable region that we would use to tell the 2 microbes apart once we have the sequences. So for PCR, we mix all of the ingredients that we need together in a tube, and then we put them through hot and cold temperature cycles. Heating DNA makes it unwind, and cooling it down again lets the primers attach to our target region so that the DNA polymerase can start copying. We go through many cycles to make copies of the DNA. The amount of copies made during each cycle increases exponentially, so that at the end of a PCR, we have hundreds of millions of copies of DNA. Once we have copies of the region of DNA that we're interested in, we purify it so that there is no other unwanted DNA. Then we sequence the copies of the DNA that we made. Sequencing happens in a similar way to PCR. Basically, we make copies of the DNA in our sample, but this time we keep track of every single nucleotide that gets added. We do this by adding nucleotides with a dye and a reversible blocking agent. This means that only one nucleotide can get added to the strand of DNA at a time. And once it's added, it blocks all other nucleotides. The dye lets us see which nucleotide gets added. Each base is marked with a different color that is detected by a tiny camera when it attaches to the DNA strand. Once that color is detected, the nucleotide is unblocked and the next nucleotide is added. By keeping track of all the colors that are detected in order, we're also keeping track of the DNA sequence. Because there are so many microbes in a community, tens of thousands of sequences must be generated for each sample to thoroughly describe all of the microbes. High-throughput sequencing allows us to do this process for lots of copies of DNA for lots of samples all at once. This is one of the high-throughput sequencers we use in the lab. Also, although DNA extraction and sequencing can be done by hand fairly easily, it can take a long time, and no one is perfectly accurate. Humans make mistakes. Therefore, in big labs like the Night Lab, where thousands of samples are processed each month, we use robots that automate these processes, making it faster and less prone to mistakes. Don't get me wrong. People still have to get the samples ready and do some of the intermediate steps, but we use as much technology as possible to improve our results and efficiency. Once we have all of the sequence data back from our samples, we then have to use the sequences to distinguish the microbes, like these microbes, from each other. We'll learn how to do that next week.